I'd like to introduce you to an organism, a slime mold, Physarum polycephalum. It's a mold with an identity crisis because it's not a mold, so let's get that straight to start with. It is one of 700 known slime molds belonging to the kingdom of the amoeba. It is a single-celled organism, a cell, that joins together with other cells to form a mass supercell to maximize its resources. So within a slime mold, you might find thousands or millions of nuclei, all sharing a cell wall, all operating as one entity. In its natural habitat, you might find the slime mold foraging in woodlands, eating rotting vegetation. But you might equally find it in research laboratories, classrooms, and even artist studios. I first came across the slime mold about 5 years ago. A microbiologist friend of mine gave me a petri dish with a little yellow blob in it and told me to go home and play with it. The only instructions I were given that it likes it dark and damp, and its favourite food is porridge oats. I'm an artist who's worked for many years with biology, with scientific processes. So living material is not uncommon for me. I've worked with plants, bacteria, cuttlefish, fruit flies. So I was keen to get my new collaborator home to see what it could do. So I took it home and I watched. I fed it a varied diet. I observed as it networked, it formed a connection between food sources. I watched it leave a trail behind it, indicating where it had been. And I noticed that when it was fed up with one petri dish, it would escape and find a better home. I captured my observations through time-lapse photography. Slime mold grows at about 1 centimetre an hour, so it's not really ideal for live viewing, unless there's some form of really extreme meditation. But through the time-lapse, I could observe some really interesting behaviours. For instance, having fed on a nice pile of oats, the slime mold goes off to explore new territories in different directions simultaneously. When it meets itself, it knows it's already there, it recognizes it's there, and instead retreats back and grows in other directions. I was quite impressed by this feat, how what was essentially just a bag of cellular slime could somehow map its territory, know itself, and move with seeming intention. I found countless scientific studies, research papers, journal articles, all citing incredible work with this one organism. I'm going to share a few of those with you. For example, a team in Hokkaido University in Japan filled a maze with slime mold. It joined together and formed a mass cell. They introduced food at 2 points, oats, of course, and it formed a connection between the food. It retracted from empty areas and dead ends. There are 4 possible routes through this maze, yet time and time again, the slime mold established the shortest and the most efficient route. Quite clever. The conclusion from their experiment was that the slime mold had a primitive form of intelligence. Another study exposed cold air at regular intervals to the slime mold. It didn't like it, doesn't like it cold, doesn't like it dry. They did this at repeat intervals, and each time the slime mold slowed down its growth in response. However, at the next interval, the researchers didn't put the cold air on, yet the slime mold slowed down in anticipation of it happening. It somehow knew that it was about the time for the cold air that it didn't like. The conclusion from their experiment was that the slime mold was able to learn. A third experiment. The slime mold was invited to explore a territory covered in oats. It fans out in a branching pattern. As it goes, each food node it finds, it forms a network connection to and keeps foraging. After 26 hours, it established quite a firm network between the different oats. Now, there's nothing remarkable in this until you learn that the center oat that it started from represents the city of Tokyo, and the surrounding oats are suburban railway stations. The slime mold had replicated the Tokyo transport network. A complex system developed over time by community dwelling, civil engineering, urban planning, what taken us well over 100 years took the slime mold just over a day. The conclusion from their experiment was that the slime mold can form efficient networks and solve the travelling salesman problem. It is a biological computer. As such, it has been mathematically modelled, algorithmically analysed. It's been sonified, replicated, simulated. World over, teams of researchers are decoding its biological principles to understand its computational rules and applying that learning to the fields of electronics, programming, and robotics. So the question is, how does this thing work? It doesn't have a central nervous system. It doesn't have a brain, yet it can perform behaviors that we associate with brain function. It can learn, it can remember, it can solve problems, it can make decisions. So where does that intelligence lie? So this is a microscopy video I shot, and it's about 100 times magnification, sped up to about 20 times. And inside the slime mold, there is a rhythmic pulsing flow. A vein-like structure carrying cellular material, nutrients, and chemical information through the cell, streaming first in one direction and then back in another. And it is this continuous, synchronous oscillation within the cell that allows it to form quite a complex understanding of its environment, but without any large-scale control center. This is where its intelligence lies. So it's not just academic researchers in universities that are interested in this organism. A few years ago, I set up SlimeOco, the Slime Mold Collective. It's an online, open, democratic network for slime mold researchers and enthusiasts to share knowledge and experimentation across disciplinary divides and across academic divides. The Slime Mold Collective membership is self-selecting. People have found the collective as the slime mold finds the oats. And it comprises of scientists and computer scientists and researchers, but also artists like me, architects, designers, writers, activists, you name it. It's a very interesting, eclectic membership. Just a few examples. An artist who paints with fluorescent physarum, a collaborative team who are combining biological and electronic design with 3D printing technologies in a workshop. Another artist who's using the slime mold as a way of engaging a community to map their area. Here, the slime mold is being used directly as a biological tool, but metaphorically as a symbol for ways of talking about social cohesion, communication, and cooperation. Other public engagement activities, I run lots of slime mold workshops, a creative way of engaging with the organism. So people are invited to come and learn about what amazing things it can do, and they design their own petri experiment, an environment for the slime mold to navigate. They can test its properties. Everybody takes home a new pet and is invited to post their results on the Slime Mold Collective. And the collective has enabled me to form collaborations with a whole array of interesting people. I've been working with filmmakers on a feature-length slime mold documentary, and I stress feature-length, which is in the final stages of edit and will be hitting your cinema screens very soon. It's also enabled me to conduct what I think is the world's first human slime mold experiment. This was part of an exhibition in Rotterdam last year. We invited people to become slime mold for half an hour. So we essentially tied people together so they were a giant cell and invited them to follow slime mold rules. You have to communicate through oscillations, no speaking. You have to operate as one entity, one mass cell, no egos. And the motivation for moving and exploring the environment is in search of food. So a chaotic shuffle ensued as this bunch of strangers, tied together with yellow ropes, wearing Being Slime Mold t-shirts, wandered through the museum park. But they had to, when they met trees, they had to reshape their connections and reform as a mass cell through not speaking. This is a ludicrous experiment in many, many ways. This isn't hypothesis-driven, not trying to demonstrate anything. But what it did provide us with a way of engaging a broad section of the public with ideas of intelligence, agency, autonomy, and provide a playful platform for discussions about the things that ensued. One of the most exciting things about this experiment was the conversation that happened afterwards. An entirely spontaneous symposium happened in the park. People talked about the human psychology, of how difficult it was to let go of their individual personalities and egos. Other people talked about bacterial communication. Each person bringing their own individual interpretation. And our conclusion from this experiment was that the people of Rotterdam were highly cooperative, especially when given beer. We didn't just give them oats, we gave them beer as well. But they weren't as efficient as the slime mold. And the slime mold, for me, it's a fascinating subject matter. It's biologically fascinating, it's computationally interesting, but it's also a symbol, a way of engaging with ideas of community, collective behavior, cooperation. A lot of my work draws on the scientific research, so this pays homage to the maze experiment, but in a different way. The slime mold is also my working material. It's a co-producer of photographs, prints, animations, participatory events. Whilst the slime mold doesn't choose to work with me exactly, it is a collaboration of sorts. I can predict certain behaviors by understanding how it operates, but I can't control it. The slime mold has the final say in the creative process. And after all, it has its own internal aesthetics. These branching patterns that we see across all forms, scales of nature, from river deltas to lightning strikes, from our own blood vessels to neural networks. There's clearly significant rules at play in this simple yet complex organism. And no matter what our disciplinary perspective or our mode of inquiry, there's a great deal that we can learn from observing and engaging with this beautiful, brainless blob. I give you Physarum polycephalum. Thank you.